There's a few things that I felt we didn't highlight enough in some of the other presentations. So I'm really going to just go through that. It's not a very exciting presentation because it's all the things we never talk about, like the PPE and the regulatory things. But I think it's necessary to just make a few comments on our experiences with some of these things. It's very much our South Africa experience. So it may be a little bit different in other countries. But I think what is good is to try and standardize some of these things for the region to avoid confusion. Even though you may get away in a country with less biosecurity or less biosafety or less PPE, I think it is important that we standardize some of these practices going forward. So I refer to these operating guidelines that we've written, and it's really extensive. It's hundreds of pages. So I've taken some of the information that I'm going to present out of this. I will put these two manuals also in the same folder with the presentations. So then you can look at it if you need to. So the additional things that I just want to mention, and I'm not going to go into it in detail, it is in our manuals, is general safety in the field. And this will depend very much where you're sampling. And then Miranda mentioned waste. So I just want to say something about the shop waste disposal and other waste disposal. Mention something about disinfection procedures, talk a little bit about PPE and the biosafety levels and the things that we need to consider there. So health and safety is really important. We talk so much about pathogens that we're only thinking about biosafety the whole time. But there's other things that we also need to consider when working with wildlife or any animals or any humans. You're going to collect samples, you're going to test it, you're going to package it, you're going to ship it somewhere. There's a whole process. It's not just when you're sitting there with a bat that there's a risk. It's also the person who needs to take those samples to a lab. It's the person who needs to ship those samples internationally. It's the person who needs to test the samples. So we really need to think about the whole process if we talk about health and safety. And these risks are very different. It's very difficult to say this is the SOP to decide on risk, because it depends very much on your geographical location, the disease outbreak status, the activity that you perform. And in a lot of cases, we don't have information. So if you tell me there's no rabies-related viruses in your country, you can't just ignore if somebody gets bitten by a bat that there's a potential of a rabies exposure, just because there's no data on the pathogen in that area. So we really sometimes need to think broader than just what we think we have in our country. So it's really about what can be in that sample, not what we know will be in that sample. Or in that animal species. We need to think about route of transmission, and rabies is probably the disease that we've studied the most in bats. There's a long history with bats, and I think before we started finding all these other pathogens, the only thing we thought about was rabies, rabies, rabies. So if we handle an animal, we were only worried about a bite. We give people pre-exposure rabies vaccinations and we think we're okay. We're not worrying about anything else, like what can we get from the fecal, what can we get from the urine, what can we get from the blood, even pathogen inhalation. If you're going to go crawl around in a cave with guano this deep, there's a lot of other things that you can also get by inhaling that. So we really need to think about the whole picture, not about just rabies anymore. So there's always a minimum amount of PPE that we need to wear. So with all of this, and we all know this, we work with these pathogens. The prevalence is so low. The titer is really low, but we can never say there's a zero risk. And that is always my message. You know, if one of us in this room work with these animals and we get sick, there's a lot of repercussions that's going to happen because of that. It's going to be bad for conservation, for disease work, for everything. So if there's little things that we can do, or sometimes they're not so little, we need to do that to try and prevent us from getting infected, somebody in that chain from getting infected, but also protecting the bats. White nose is a very good example of what can happen if we're also not protecting the bats when we move around between caves and other areas. So think about the bigger picture, not just about the inconvenience of trying to implement some of these things. It isn't nice working at 40 degrees with full PPE. It isn't. I can promise you that. I had to do that a lot of times in my life. But think about the bigger picture and what the risk can be if we don't do that. So there's always, this is our medical requirements. It might differ. But one thing that's becoming really important if you go and work, especially in remote areas, is your health insurance. If you need to be able to get out of there and you don't have health insurance, it's really important. So it's always something you need to think about if people come and visit you from other countries, because they won't necessarily have the same cover that you have in country. So make sure that your people are covered, that's in your team. We look at lung function and chest x-ray, specifically because we wear respirators regularly. So having a respirator on puts some pressure on breathing. So if you already have somebody that's got some conditions, it may get worse. And this is really if you do it a lot, but we do it a lot. So we really try and do that. You always need to be vaccinated. Pre-exposure vaccination for rabies is essential. And all those guidelines are on the WHO site. We also do tetanus, and then we also do hepatitis A and B, which I think is optional, but it's good if you can do that one. And then there's obviously, with time, there's different vaccines that you may also need. COVID's one of them. So we start seeing the COVID vaccine as an occupational vaccine because we work with coronaviruses, so not just because of the current pandemic. And then we sometimes forget about anti-malaria prophylaxis. We really need to think about that if we go into areas where malaria is endemic, which is basically the whole of Africa and our northern areas in South Africa. So just a few things that you can consider. Then I just want to show a few photos. So what is the environments that we work in? And you need to think about these beforehand. You don't want to stand in front of an elephant and didn't think there's big five in the area. You know, you really need to think what you're going to deal with. You need to think about snakes. And this is actually not a snake that's going to kill you. There's others that, or I can tell you, that's more poisonous. But the areas that we work in, there's always snakes. So you need to have a plan, because otherwise if it happens, then it's just chaos and panic. So if you have a plan beforehand and it's communicated with everyone, you know what to do. And usually you mitigate the risk. There's all kinds of wildlife, there's water, there can be floods, there can be fire. So you really need to think what can happen in your area and just have a discussion with your team and say, listen, this is what we do in this situation. This is what we do in this situation. Planning helps in how you react. You can never really get rid of risks. I mean, you can't get rid of all the snakes. You can't get rid of the big five in an area where you're working. You can't get rid of a flood. But if you plan, you usually mitigate at least your reaction. So this is the type of environments that we work in, and it's different. I mean, if you're going to catch a bat from a tree versus climbing into a cave or climbing on top of a roof, there's different types of things that you need to think of. You can slip and fall off a roof. Or if you need to climb around in a cave, you can bump your head, so you need a hard hat. If you go into mines, you need to consider things like, is there enough oxygen? You can't just go into those areas. So it's really things that you need to think of beforehand. And this is just a few examples of places that we crawl into and go into that you need to consider. So know your environment. And then working with the animal itself. You get different bat species, as everybody told you, and they also have different personalities. Some of them are really just nice to work with, and they don't bite. They will let you take the samples, and there they fly off. Others are not. You do get bats that's just not in a good mood. And then there is the risk of getting bitten. And obviously, you need to have the right gloves on. You need to be trained well. You need to be able to handle it if you do get bitten. What's the procedure? It shouldn't be panic at that moment. We need to know what to do. So one of the things that I always say is training, training, training. usually, and then also how you plan your studies. If people get tired, if you work too many hours, that's when accidents happen. So you really need to think about how you can make these risks less. I show this, and you've seen it a lot, and we will also set it up for you tonight. But if you look at this table, there's a few extra things that you need to think about. We work with power supplies. So what's going to happen if it starts raining and everything gets wet? Additional risks of getting shocked and electricity issues. You know, so we really need to look at things like that. We talked about waste disposal. It doesn't need to be fancy systems, but you need to have something where your sharps go. And then what are you going to do with that container? It's not just having the container on the table, and now you pack up, where does it go? Because you can't go and throw that in normal waste disposal sites. So you need to have a plan where you're going to go with those containers. We, for instance, bring it back to Pretoria, and our waste disposal companies get rid of it. Other places in Africa where you don't have that, it makes sense sometimes to just have an agreement with one of the hospitals, because they do have sharps, and you can drop it there and it goes into their disposal systems. Your other waste that's not sharps, it's still bio-waste. You can't just go and throw it away. So it needs to go into a system. One of the things that we sometimes say we can do is burn it. Obviously, that's not a very environmental friendly way. So that shouldn't be the first option. It's really if you don't have any other options that you can maybe look at that. So we mentioned necropsies. So there's a lot of things you can do to be safe while you're collecting things like fecal urine. You can put it directly in an inactivation buffer. But as soon as you start cutting open a bat, there's a lot of additional risk. So you're now working with an animal where there's blood all over, there's tissue all over. So immediately there you need to think about additional PPE. You're also creating a lot of waste that you need to do something with. What are you going to do with a carcass? You can't just throw it somewhere. We put everything in formalin and it goes to the museum, but you need to have a system to do that. So what I want to show you, so for instance, there's ways of inactivating samples, and carcasses is a good example. While you're doing the necropsy, it's obviously, you need to consider everything as infectious. But if you do put it into the formalin, it's completely inactivated. So that sample you can then take somewhere. So in our case, we try and do that in the field, because I don't want to carry a bat carcass 400 kilometers in a car that's not inactivated. So think about the risk and where in the process you can mitigate that risk. Yes, then needles. And this is usually, remember, you can't do this work completely on your own. So it's not just you sitting at the table. There's somebody there, there's people bringing bats, and there's needles. So you really need to think about how you work with needles, with bleeding. Tattoo guns, you don't really want to tattoo your fellow partner. Pit tags, you will see, has got quite a needle on it. You really need to look. So needles is also really serious exposures, because it breaks the skin. Sometimes it's contaminated already because it was used on a bat. So we really need to think, you never just hand somebody a device with a needle showing towards them. So there's ways of handling needles. So again, coming back to planning and good preparation is really instrumental in minimizing risk. Can't get completely rid of it, but if you plan for it, then there's a good chance that it will be minimized. So you really need to think about it, identify it beforehand, evaluate it, think about a way to mitigate it, involve everybody in the conversation, and make sure they understand it. Just handing people a paper SOP doesn't work, in my opinion. You know, you need to do some training, you need to maybe do some exercises that's practical for people to really understand it. But there needs to be conversation on these things also. And then monitor the outcome. What we do a lot, because we also have high biocontainment labs, if we do have an accident, we need to rethink about, is our SOPs good, even in the field. If something happens, maybe there's something wrong in the way we communicate it or the way that we implemented. So then you need to change it and revise it. So I've talked about all the potential hazards and risks, so I'm not going to go into that again, but it is weather, terrain, toxins, health conditions of people. Allergies is a good example of that sometimes pops up in the field and you're in a remote area. So you need to know those type of things from the people that's involved. And then there's biosafety that I will talk a bit more about. So I'll just show this picture. So in terms of waste management, sharps need to go into a dedicated container and there needs to be a process for safe disposal. All other waste into bio-waste bags, and burning is not really an option. I would actually like to hear, if you have your discussions this afternoon, how do you deal with bio-waste? Is it an easy process or not? Because I think it is actually a challenge in a lot of countries on what you need to do with your bio-waste and doing it safely. So there's ways that if we look at biosafety, there's a few points that you can look at to try and assess it and to try and decide what PPE you should use. And a lot of this is also used to decide what level we should handle these pathogens if they are infectious. And this is not clear-cut. It's actually quite difficult. Jacqueline can maybe comment on that because she sits on the South African committees, I think, trying to decide on these things. And it's really difficult to sometimes decide what needs to be handled in a specific way. But if we think about animals, we need to think about species. But again, you usually make these decisions on what you know and what else can be in there. So that's always a question that you need to ask yourself. What pathogens are likely to be present? Very difficult one to answer sometimes. Pathogenicity, this is obviously things that we know. Can it be transmitted to people? Can it be transmitted to other animals? How pathogenic is it? Also a difficult one. We don't know this for all our pathogens. Potential exposure opportunities. So what's the route of infection? What are you planning to do with this? This is typically, so if you're collecting a sample from a bat infield, let's use that example. You're doing a swap, you're putting it in an inactivation buffer immediately, and you're shipping it to a lab, and you're just going to extract RNA. That's a very different risk from taking that same sample, freezing it, just freezing it, no inactivation buffer, taking it to your lab, and you put it in cell culture. Now you want to grow up something. You know, so you really need to think what you're going to do. So the risk is different depending on what you're going to do, and also how you originally collected that sample. Is it inactivated or not? So those are the type of things that you need to look at. Infectious dose and stability of pathogens in environment. We know that for some pathogens, not for all of them. What do we know from the literature, from animal studies? And then prophylaxis and treatment. That's usually one that we know. So like with rabies, we know we can give pre-exposure, we know we can follow up after a bite, or we know there's nothing that we can give people. Most of the time with the type of things we work with, there's no prophylaxis with treatment. So that option is normally not there. So one of the things we've been discussing quite a bit in South Africa is there high-risk species? And I'm still asking that question. You know, can we say, if somebody asked to work with rosettas, for instance, is it a higher risk species than somebody asking to work with mops? So is there a higher or a lower risk in that situation? And we at some point flagged rosettas as a high-risk species. There was one reason was it's a cave-dwelling species, high population density, so immediately there's a lot of them if you go into that cave. And if you look at this map, it's been associated with a lot of different viruses, including filovirus. We know that. We know rosettas is a reservoir host for Marburg virus. So immediately, if you work with rosettas, we need to be careful. If we can say other species are lower risk, I'm not so sure. I think we can debate that. But rosettas, for instance, we know is higher risk at the moment. So what we decided on in terms of PPE, and this was after a lot of discussions, we came up with this table. where we've got a certain activity and then a certain type of PPE that needs to be used. So if you're doing mist netting, harp trapping, and hand netting, or collection of any biological or genetic material from animals, it's double-layered gloves. That's actually a given always. You always just wear double-layered. And this is, we say nitrile, but some people still use latex or so it's any of these surgical gloves. An N95 respirator, and there's different types that you can choose from. Face shield or goggles, and then dedicated long sleeve field clothing that you specifically use for bat work. And we like using our gumboots. And I remember, Mark, we had this discussion that you can't always wear gumboots because some surfaces are really slippery. So gumboots is not a good type of shoe to try and climb around in. But the idea here is that it's dedicated because you need to disinfect. So it needs to be things that you're willing to disinfect. If it's your very expensive hiking boots and you don't want to spray it with bleach, it's going to be an issue because it's contaminated. So it really needs to be things that you're willing to disinfect. Necropsies, disposable gown over everything, because you want to throw that away because you're going to work with blood and all of that. You don't want something that you're just going to spray down. Work in the roofs of houses or roosts. This is a difficult one because if you look at this type of setup that we have here, if you go into very narrow spaces, you really battle to crawl around with all of that. Lau can comment on that. You sometimes get stuck. So in this case, you can sometimes just wear an N95 respirator mask and a face shield or goggles and your gumboots. And then small roosts. We said double-layer nitrile gloves, bite-proof gloves, N95 respirator, your goggles or face shield, again, dedicated field clothes, disposable coverall, and gumboots. The reason why we want these disposable coveralls, or at least something that you can spray down completely like raincoats, or because you're going into these roosts. So you're getting full of fecal, full of urine. You can't just come out there with your normal clothes and spray yourself because it's actually going to go through everything onto your skin and everything. You want a second layer that you can take off because you're not going to take those clothes off because then you're going to be without clothes. So you really need an extra layer that you can have on top of that. And then our agreement at the moment is with high-risk roosts, which is specifically our fruit bats, disposable coveralls, full-face respirators in this case. To make our life easier, we just work in the same thing the whole time. But you can use less PPE in certain situations. So I just want to show a few different examples of N95 respirators. So this is not your COVID mask that you buy at the pharmacy, although these days I think you can actually buy N95s at the pharmacy. What's difficult with them, it's actually supposed to be fit tested. It's not a one-size-fits-all. So if it doesn't fit you properly, it's not protecting you. So the complication, if you really do this correct with N95 face masks, you need to fit test every individual, and you need to make sure it fits for it to be really protective. And that's why we moved away from it. The other thing is you need to throw it away. So you need to dispose of it. You can't reuse it. There's actually training on how to remove it. And this sounds maybe like a weird comment, but we had issues in South Africa where people got histoplasmosis because of the way they remove their masks. Because they come out of the case, and you're sweaty and you can't breathe, and they just pull it off and it goes all over their face. You know, there's no disinfection. You can't just pull it off, and then it touches the rest of your face. So there's all kinds of issues using it. It's possible. The other issue is it's not covering your face, so all your mucosal membranes are open, your eyes specifically, so you need to wear additional eye protection and face protection if you wear this, because otherwise the urine and fecal and everything will be all over. So this is just examples. This is another option that you can wear, that, or this is the ones we wear. You can wear that with goggle type of things, or you can wear the full respirators, or now I can't remember what we called these. It's the 3M face masks, but they're not as expensive as the full face respirators, and they're actually quite a good option because it closes the whole face and it's an N95 protection. So just in terms of costs, because we wanted to look at costs, because that's always the issue. So if you look at the full ones, a kit like this will work out $1,600 US, and you have some disposables that's about $10 US. This actually now became more expensive with COVID. It's probably 3 times as much because all disposable PPE went up a lot. This will cost you about $40 US, but you're gonna not be able to reuse it. This you reuse a lot of it. And then this one's is the other option, is about $260 US. And you can at least use the respirator part of it. So that's the options in terms of what PPE will cost you. Then I just want to say something about disinfectants. So it's important that you understand disinfectants, and that whatever you use, you know it's going to be at the correct concentration, and you know the time that's needed for it to be effective. And this is different for different disinfectants. So you can buy the veterinary disinfectants like the F10s. What is the contact time with F10? 30 minutes. So if you use that in the field, you need to stand here and wait until that time is gone before you can wipe it off or you can pack your things away. Ethanol, 70%, it doesn't even work against things like spores and all your viruses, so it's anyway not a good option. 10 minutes. Then Virkon is one of the things that we use in the lab, expensive, it's not cheap. It's also 10 minutes at 1%, it works against everything. The cheapest and the most effective is actually just bleach. You can get it anywhere, it's no special bleach. You just need to make sure of the concentration. It will inactivate everything. The only problem with that is it's hard on your equipment. So a lot of times you need to spray and you need to wipe again with water or with ethanol. Very hard on your clothes. So you always have clothes full of bleach. But you can just wear your older clothes for that. So just make sure you understand disinfectants. And disinfectants must be used at all points. Your bat bags that you've hanged the bats in after you've processed them, there was bats in them, needs to be disinfected. Your table, your clothes, walking into a cave and out, your boots. So we usually just put a scuttle full of bleach where you can wash your shoes. But everything that got into contact with the process needs to be disinfected. If you spin down or centrifuge your blood at the end of the evening and you're going to put your tubes away, it needs to be sprayed before it goes into a container. So it's really everything. Then just getting to some of the permits and legislation, you can see how this list is looking, and it's growing every day. There's just so many things that we need to get in place before we can do a bat study. We want to move samples around. There's a lot of issues. So for instance, in South Africa, this is what we need. Our veterinary services have an approval process for all work done on potential animal diseases, which we call Section 20. A lot of the other countries is also implementing that. We can't do any work if we don't have this. You only get this if you can do the work in the right biosafety facility. So they really look at what's the risk in terms of biosafety and biosecurity. So storing of samples is also for them extremely important, and how we trace them. So for instance, if I'm going to work with infectious bat samples in South Africa, it needs to be BSL-3 or even BSL-4 if there's potential filoviruses. There's no way around it. The only way around that is inactivating your samples in the field. You've got inactivated samples, and you can then work in another lab with it. So those are your considerations depending on your country's regulations. The same for bringing in or shipping out samples. Can't bring in samples that's infectious into a BSL-2 lab. It immediately needs to go to a higher level. Ethical approval, John spoke about that. Ava spoke about that. One thing we're seeing with the ethical committees, it's not just, John made the comment that it's about working on live samples. It's not the case with our ethical committees anymore. They're also looking at the biosafety aspects now. So it's even inactivated retrospective samples. They look at everything. So it's becoming more and more difficult to get approvals. And there's more and more things that we need to answer. In South Africa, we've got provinces. We need to get a permit for that. If you enter a cave, there's another permit for that. It's not all under the same permit. Then permission from landowners, municipalities we need, traditional leadership, transport between provinces, we need another permit if we take it out of a province. So if you work in different provinces, you need to get that. And then if there's World Heritage or national parks, there's additional permits on top of that you need to get from those sites. And then the approved laboratories. And the laboratories get approved by the government. So that's also a process that you need to go through, a long process. So if your lab is not already approved, you're not going to start a study soon. You're going to really have to first get that in place. Then there's other things that's becoming more prominent. Site test has been there for a while. I think Ara mentioned that also, that you need to know the conservation status. The Nagoya Protocol is becoming, I made a spelling mistake there, becoming very difficult. I don't know if you're aware of that protocol. It's applicable to all countries that signed the agreement, so it's basically all countries, where it's really looking at the use of genetic material and sharing of information. So any biological sample falls under that. And your country may not approve that you remove the samples from your country without a process to apply for this and the permissions. So there your collaborator can't do anything. You know, it's really, your country is going to say, I need this approval. So please look at those things beforehand. We couldn't get samples out of African countries lately because of Nagoya protocols. And no process to apply. So it's there, but there's not a good process to apply and get permission. So it becomes really tricky. Export permits are needed from some countries. So although you have an import permit into another country, you still need to get an export permit to get it out. And then all the human studies, there's additional approvals for human studies that Ava covered quite well. So just don't let this put you off, because I think most people is like, I'm gonna stop working on bats. If you went through this process once, it takes you a while, you know what they want, you've sorted it out, and then it goes much smoother after that, with a few hiccups here and there. But this is the reality that we need to deal with. Then I'm just going to quickly go through cold chain and safe sample transport. So cold chain is extremely important to think about at the beginning. And if you can't maintain a cold chain, we need to look at alternatives of how you sample. So cold chain means that from the moment you take that sample in the field, it needs to go, for instance, into liquid nitrogen or something that's going to really freeze it if you want to look at viruses. Then it needs to stay in that process till it gets into the lab, and you need to put it in a -80 if you want to work on those samples and the integrity is in place. If you can't do that, you need to look at alternatives. You need to use buffers that will protect your RNA of your viruses, and the RNA-DNA shield is one of them, and you can then do room temperature. So you need to think about that, because if you break the cold chain without having one of those buffers in your samples, your testing is going to be really bad quality. So then you've put all that effort in, and you don't have good quality samples. And this is a big issue in most of our African countries, even in South Africa, where you don't have stable power supply for -80 freezers. You don't even have a -80 freezer. So we really then need to look at alternative ways of sampling and putting it in a buffer where it's protected. Then there's also quite a lot of regulations in terms of transport, and that's international regulations, how it should be packaged. All of that needs to be adhered to. Inactivation, this was mentioned, so I'm going to be really quick on this. We currently use DNA or RNA shield as a buffer of inactivation. A lot of people used RNA later before. It's now been proven that samples are not completely inactivated in RNA later. So that's not an acceptable way of inactivating samples. DNA RNA shield, you can also do at room temperature, so it's fine. The samples doesn't need to be kept cold, and it's inactivated. So immediately you get away from all the biosafety issues in terms of processing the sample, not in terms of how you're going to catch the bat and put the sample in the tube. And then blood, we inactivate at 56 for 30 minutes, which is the approved CDC method for most pathogens. And then that's also inactivated. The problem with blood and serum, you still need a cold chain. So you're not going to get away from that. So that's usually quite a problem with the African studies. And then voucher specimens we put directly into formalin after necropsies, and then that can go to museums safely. And there's no issue of biosafety. The last thing I just want to mention, and Miranda mentioned it, is databases. Your data needs to be really, really organized, and you need to have a system where you check this. Otherwise, by the time you want to look at the results, you realize it's chaos. And this needs to be student-proof, research assistant-proof, field assistant-proof, professor-proof. It really needs to be a system that somebody checks. And we've learned that lesson a few times in my group, and we really need to make sure that our databases are perfect. So we moved away from Excels to these Airtable online databases where you can also track who entered what. We can check data integrity. You can share that online between collaborators. You can have different levels that you share it with. You can share it with the government, so you immediately have your reporting systems. So it's really something that we invested with together with EcoHealth, and we really developed one database where everything goes into. Even the people's medical records, everything is in one place. And it's linked to our results and all our PDFs, our photos, everything is there. So this is something we developed, and we can share it with other groups as we go forward. So reporting, discuss your reporting plan at the start of the project. So if we talk about who should be around the table when we start a project like this, your government department should be there from the beginning. Because if it gets to reporting with these diseases, you can't just as a researcher go and say, I've got Ebola in a bat in Pretoria or somewhere. That's going to create a lot of issues, and it's actually against our reporting guidelines in South Africa. So you need to know how you're going to deal with a positive before it's there. And then really look at your country reporting procedures, especially in terms of notifiable diseases. That's usually where a lot of the issues comes in, because we report to OIE and WHO on those. Be responsible. Sometimes I think it's more about the excitement of finding something new, and everybody tells everybody, and the next moment it's chaos. Really think about the implications for bats, for humans, for animals before you report it, and how you're going to do it responsibly. And it needs to be in full partnership with the authorities. They need to tick off what we report on before we report on things that's going to create a lot of other issues.